body, and it is about 3.2. Can you, sir, this you can remove this one because this is coming to me, wetting and everything. Can any other person can handle it? So it is about 3.2 billion letters of our DNA in our body. So it is huge data. When you compiled it in form of paper, it will be about 61 meter high and about 200 telephone directories you can fill with. So one sequenced genome equals to 2 billion bytes and or about 200 gigabytes. So that is enough to fill the memory of our average laptop. So you can imagine how big it is, how this data is containing too much. And now this all human beings are 99.9% bases are the same. Only remaining 0.1%, that is about 3 million bases that makes a person unique. Unique in sense their attributes, characteristics, their traits, how a person looks, disease he or she develops. These all are about only 0.1%. So you just imagine how much we are similar in the DNA concern. And this variation, when we see, these are harmless type or harmful or latent type of variations. Harmless in the sense, you can say there is some change in phenotypes only, some characteristics are changed. But harmful, when you can say certain diseases like diabetes, cancer, heart disease, Huntington disease or hemophilia, or in latent in such cases like they are found in coding and regulatory reasons. They are not harmful on their own, and the changes in each gene only becomes apparent under certain condition. When they got certain situations, then the person is susceptible to particular disease. For example, lung cancers, everyone is susceptible to it, but those who are smokers or those who are consuming particular kind of food, they are more susceptible. So that latent thing is coming to the particular kind, and that person is suffered from that particular disease. Now pharmacogenomics and pharmacogenetics. So genomics is the study of entire set of genetic instructions found in cell. That science is known as genomics. And pharmacogenetics, in short, you can say PGT. This is the science for in which you study about the genetic variation of drug responses. And in pharmacogenomics, that is the application of genomics to the study of human variability in drug response, and application of genome-wide SNPs, singular nucleotide polymorphism, these scans, and the causes gene expression analysis through ACE. When you say genome, genotype or phenotype means, genotype means that is the genetic traits or reliabilities, or you can say genetic makeup of organism. And phenotype, when you say some organisms observable traits. So pharmacogenetics and pharmacogenomics, these are the terms, and these are the, you can say, real words which are making this personalized medicines useful, because these are the play important role in development of better medicines for populations and targeted therapies with improved benefit risk for individuals. And that is very important for made to order drugs, because what we are studying in this, so how we can make particular kind of drugs for particular patient or particular subpopulations. This is for that. And this shows the how gene determines individual variability to drug response. And for pharmacist, it would easy to predict patient may respond to how patient may respond to drug, which is help for genetic. So there are different type of prediction, you can say, whether who will benefited and who will not respond at all, and some experiences, or you can say some adverse reactions are there. So different type of prediction is possible with help of this Then this employment of gene therapy as experimental technique that uses genes to treat or prevent disease. As you know, everywhere nowadays, this gene therapy is available for many dreadful diseases or some rare diseases. So this gene therapy provide some very tremendous roles in treatment, diagnosis, and these are given different kind, like replacing a mutated gene. We can change that mutated gene, and because that is causing diseases, and we replacing with the healthy copy of that gene, or inactivating it or knocking out that is mutated gene that is functioning improperly. We can replace it, we can knock out it, and we can change with the healthy one, and introducing a new gene into the body to help fight a disease. We can also introduce you can say better, or you can say new gene. And with help of better drug discovery and pharmacogenetic, and with help of pharmacogenetics and pharmacogenomics, we have better drug discovery that will increase drug efficacy, eliminate adverse drug reactions, that ensure success for drug approval in clinical trials, and increase efficiency of clinical trials. So you just see this picture is telling us about the yesterday's medicines and tomorrow's medicines, and what we are using just now. So in yesterday's medicine, we are providing same diagnosis, same prescription for all patient groups. So you can say this is average kind of patients, and we are giving same. And there are response like if drug is toxic but beneficial, drug toxic but not beneficial, drug not toxic and beneficial, and sometimes drug not toxic and not beneficial. These four type of categories is possible. Now the journey of precision medicines, or this shows that today we are using most medical based on the expected response, or you can say average patient response. But in with help of precision medicine or personalized medicine, medical care based on individual genomic, environmental, and lifestyle differences that enable more precise way to prevent and treat diseases. Okay. So in the role of the new medicines, just a minute. So route to the new medicine is a long and expensive one. At present, whatever route we are using for discovering of medicine, that is very long. It required at least 10 to 15 years to discover one particular drug. And when you got one idea and that you transform into the viable product into the market, it required at least 11 to 15 years. And it cost about, average cost is about 800 million to get a drug to the market. And in some cases like anti-cancer drug or AIDS drugs, it is more than 800 million. It is around $1.2 billion even required. And pre-screening should allow clinical trials to be smaller, faster, and less expensive. Therefore, the consumer could benefit in reducing drug cost. So today, response of drug is only 30 to 60%, and does not respond to drug is everyone else is maybe not response at all. But in with help of personalized medicine for tomorrow, response to drug will be about 100%, and does not respond to drug is about 0 only, because only we will use only responders, not other persons, not average person. We will choose only responders, so that 100% response to the drug to that particular patient. So this 30 to 60% will increase to 100% response, and drug will use effectively. 100% cure will available for the patients. And human genome diversity is, just a minute. So in human genome diversity, use SNPs. I already told you SNPs, single nucleotide polymorphism, or simply you can understand that this is DNA sequence, and this is change when we change only one alphabet, or by mutation is only one alphabet is changed. So there is a variability occur, and so some could have a profound change in the meaning or function, or other may minimal or no effect. If there is one alphabet is changed, sometimes there is no effect, but sometimes it is you can say drastic change is there. For example, just see the English meaning kettle and battle. There is only one letter is different, but their meaning is totally different. But in case of tumor and tumor, spelling is different, but their meaning is same. Similarly, in case of DNA sequencing also, when there is some changes, so these SNPs are very powerful tool. These help in prescription, right drug, at the right dose, at the right patient. Okay, and this understanding SNPs that has shows promise for improving diseases detection and treatment. When we understand this SNP, we can improving disease diagnosis, their detection, their treatment, and this arises consequences of SNPs arises because of the consequences of the mistakes during normal DNA replication. And average frequency is 1 in 1,000 base pair. There may be chances of one SNP will occur there. And in 94 general population, there is no SNP, but in 6% population, there is possibility of SNP is there. Example of SNPs like in sickle cell anemia, there is beta globulin is there. And in for breast cancer, there is SNP is predisposed of BRCA1 gene. This is the faulty gene which causes breast cancer. Okay, and this SNP can change drug responses through the pharmacokinetic and pharmacodynamic changes. So now we come on about the personalized medicines. Because at present we are using trial and error type method of drug discovery, and that is the one size fits all, but that is not the right type of treatment for the all people, because that is not the right dose. So one size doesn't fit all. Every year in America, 2 million people die. Even in India, more than 2 million people die because of the adverse drug effects. And drug efficacy is only 60% of the population. Means you are giving 100 people some drug and only 60% population is giving response to that particular drug. Otherwise, they are just giving the adverse drug effects. And about 40% of poor drug effects or no effects at all. Okay, so patient with same diagnosis, some are predicted good response to tested drug, some predicted poor or non-response, use different drug, or they are using some different drugs, and predicted increased toxicity risk. So what we do, because percentage of patient population for which particular drug is a class is ineffective or an average. For example, certain class of drugs like antidepressant or asthma, diabetes, arthritis, Alzheimer's drugs, cancer drugs. So their effectiveness is very low. In case of cancer drug, only 75% drugs are ineffective, whatever we are giving on an average, or one size fits all type of drug, or average population type of drugs we are providing them. So in such cases like cancer or Alzheimer's or arthritis or very rare diseases kind of, this is not the right case. So what is the answer? So answer is personalized medicine, or that is also known as pharmacogenomics, precision medicines, individualized medicines, or stratified medicines, or P format. These are the synonyms for that. This is the emerging approach for disease treatment and prevention. that takes into account individual variability in genes, environment, and lifestyle of patient. So P4 medicines, this is also known as, it means this is personalized, preventive, predictive, and participatory type of medicines. Because average medicine, as we know, at present we are using, that is everybody's therapy is nobody's therapy, or everybody's treatment is nobody's treatment, you can say. So we wanted some tailored kind of treatment, some approaches which are useful for an individual patient. Okay. And this term, personalized medicine, is not, you can say, not very new. It was used from time beings and from ancient time also. And first time, but this was coming to light by the scientist Sir Archibald Gerard. But we are using in some or other sense from the Hippocrates time. And this, like blood transfusion, you know, so it is so particular for individual. Similarly, this personalized medicines also. And when human genome projects come into existence from 1990 to 2003 for 13 years, and that time it cost around $3 billion. Now we have complete genome sequences are with us, and now it is a very not very costly, around $1,000 we can do this sequencing. And many people, many countries have these facilities, and even in India we can do that. And time is also required less now. It is not required 13 years to do sequencing now. It requires some days only. And personalized medicine is rapidly evolving field in which physicians use diagnostic tests to determine which medical treatment will work best for each patient, or use medical interventions to alter molecular mechanism, often genetic, that causes diseases or influence patient response to certain treatment. And by combining molecular data with individuals' medical history, circumstances, and values, healthcare provider can develop targeted treatment and prevention plans. And differential diagnosis is also available with help of predictive biomarkers that test based on demographies, patient history, physical examination, and certain information like age, sex, ethnic groups. These are valuable for that. And several relative newcomers like advanced imaging technologies available nowadays, magnetic resonance imaging or CT, and these omics techniques. With the help of that, this personalized medicine works more better. And the mechanism for this personalized medicine is natural variations, DNA polymorphism. Because of this DNA polymorphism, this natural variation is occurs, play a role in risk of getting or not getting certain diseases. And different external factors are also there, like environment, diet, exercise, along with polymorphism, also determine the individual's risk for disease. And natural genetic variations determine drug efficacy, and variation in DNA that lead to differences in pharmacodynamics and pharmacokinetics in individual patient. And this based on patient individual genetic expression. Some biomarkers are there, DNA microarrays, they can detect, and they help in finding the best medication for the patient. And what makes it possible? This human genome sequencing, new technologies facilitate handling of the data, lot of investment in public and private sectors, and future of medicines. This personalized medicine is the future of medicines. As you know, the case of Angelina Jolie, this very famous Hollywood actress, when she come to know that she is having some faulty gene in her body, she go for a double mastectomy, and she initially she was having 87% chances of getting breast cancer because of that faulty gene, but now she's only having 5% chances. Why it is so? Because now diagnostic testing is possible, appropriate optimal therapies are possible, and genetic content and molecular cellular analysis is possible. We come to know that after certain period of time or certain years, you are going to suffering from this kind of diseases. So you take the initiative and you can save yourself. Now Angelina is only just 5% chances to suffering from this breast cancer because she went for double mastectomy. So this personalized medicine is the answer for this kind of future diseases also. And Nature, Time magazine, HPR magazine, they also give their frontline story about personalized medicines. If we want to offer the right drug to the right patient for the right disease at the right time with the right doses, and generally metabolic data that allow drugs to let tailored to patient subgroups, because with the help of this genetic and metabolic data, we allow drugs to tailored as patient subgroups available. Now, what are different aims and scopes of personalized medicines? As personalized medicines translates recent genomic, genetic, and omic advances into the clinical context. And these players involved in this personalized medicines upgradation or uses, that is academic and clinical researchers, pharmaceutical companies, regulatory authorities, healthcare management organizations, patient organizations, and other in the healthcare community. This personalized medicine assists these parties to shape the future of medicine by providing a platform for expert commentary and analysis. They gives the expert commentary with the help of all these people and all these gadgets available, and the analysis is perfect analysis is done with the help of that. And these are certain points for the scope, different fields where we can implement personalized medicines. That is like implementation of genomics in medicines in cutting edge field, and advance in molecular diagnostics, biochip or microarray technologies. With the help of biochip or microarray, we can find out the faulty genes and we can remove them, and we can prepare better medicines with the help of this microarray technology. Impact of precision medicine strategy on drug development, on healthcare delivery, and developing the evidence basis for the adoption of precision medicine. I think you come to know about evidence-based medicines. This is very common word nowadays. That is also part of it. And the national sequencing programs. This is also performing by every country. In India also, we are planning for this Indian Genome Project. And in England, Genetics England program is there. We are going to having sequencing of about 1 lakh people of their all genetics and what kind of traits and every time of possibility of diseases. In India also in different phases. There will be one slide will come in front that we will discuss about that. And impact of the latest concept of development of precision medicine that is based on pharmacogenomics, pharmacogenetics, and pharmacoproteomics. So these are different scopes in that also. After that, integration of diagnosis with therapy, the use of companion and complementary diagnostics to discern biomarker statuses to guide drug users, and the prediction of adverse reactions of drugs and drug safety, treatment monitoring, And therapeutic application of genome editing and associated technologies. Ethical, legal, and social issues are there. Educational strategy and workforce development. Information technology strategies. Participant and patient engagement. Data science and data analytics. In this big data is there, and EMI, electronic data management, or digital health, cost benefit and economic outcomes. So these are certain scope of the personalized medicine in future. So we can go one by one. So ongoing research on cytochrome P450, because cytochrome P450 is a very important enzyme, or we can say superfamily of enzymes. And these are important drug metabolizing enzymes. Bioactivation and metabolism of approximately 75% drugs give cytochrome prominence in pharmacogenetic research. So study on this cytochrome 450 and their subfamilies is very important when choose particular kind of drug, their metabolites, because they gives a very good information into the diagnosis, disease, and prescription of drugs. And these are some specific examples of personalized medicines or personalized healthcare, like different type of tests, diagnostic tests are available for particular kind of diseases, some tests for particular kind of faulty genes, and what are their different functions and their implications. So disease susceptibility test, that is for breast cancer, and test for BRCA1 gene. And function of this BRCA1 and BRCA2 gene is individual with deleterious BRCA1 or BRCA2 mutations that are at increased risk of breast and ovarian cancer. Those who are affected, or those who are having this kind of gene in their body, means they are more susceptible to breast cancer or ovarian cancer. Implication is surveillance or risk modification, chemo prevention, prophylactic surgery. As I told you about the example of Angelina Jolie, she also, because of she come to know about this thing. So risk modification is best now. Chemo prevention, to provide some chemical treatment, and prophylactic surgery is possible. Similarly, prognostic test in case of breast cancer, MemmaPrints is available. That test predict the risk of cancer occurrence within 5 to 10 years after the initial event. And adjuvant chemotherapy, we should go for it or not, we come to know about this particular MemmaPrint. And a companion diagnostic effectiveness oriented for breast cancer, that is HER2 test. And for trastuzumab, this is a particular drug, Herceptin. This is known as Herceptin, brand name. And this is beneficial only for tumors with those having HER2 overexpression. Those who are showing some overexpression on HER2. So for those, you can decide if we should give them trastuzumab drug or not. If that is not having any HER2 overexpression, means there is no need to go for this particular drug. Okay, this is monoclonal antibody. So we are not going for that particular drug if they are not expressing this particular test. Similarly, there are so many examples. One is for epilepsy, that is containing HLA-B1502. This patient, if they are having this particular gene, they are more likely to have dangerous skin reactions. When you are going for carbamazepine therapy, if we giving this carbamazepine therapy to those patients, they are expressing HLA-B1502 faulty gene in their body, so they will show some skin reactions, very severe skin reactions. So you can have that decision that we should not go for carbamazepine therapy in those particular patients. Okay. And some arterial fibrillation and other indication for warfarin, other coumarin derivatives, we can test for cytochrome 2C9 or BK0RC1. So these are optimal maintenance dose of coumarin therapy is partly dependent on this cytochrome 2C9 or VKORC1 genotypes. So we can decide about the warfarin doses. So these are certain examples for which we can take decisions whether we implement this particular treatment or not. Now, certain milestones in personalized medicines are there. Approval of 11 personalized medicines representing more than 20% of all newly approved therapeutic molecular entities in last 6 years. It means this is showing that FDA is looking for more and more personalized medicine for every year that is coming. So recently, 11 personalized medicines introduced already, and approval of gene therapy is there that gives patient with special muscular atrophy the first treatment to address the root cause of their disease. Okay, so gene therapy is for those patients who are suffering from spinal muscular atrophy. So that is the best treatment now. And approval of small interfering RNA-based therapy, siRNA, you know about this. This is also very current topic and very important. And this gives patient acute hepatic porphyria. That is the first line treatment, root causes of this particular disease. siRNA-based therapy, we provide them. And clearance of approval of 7 companion or complementary in vitro diagnostic tests that can inform targeted therapeutic decisions to improve the safety and efficacy of treatment regimens. And qualification of first digital technology, softwares are available. They are designed to accelerate progress in personalized medicines. One example of that particular software, like OCX CDE software, that module is the first biomarker test for the brain injury. So this, with the help of this particular software, we can go for therapy, and short treatment is possible with help of this. Now, certain potential benefits of personalized medicine. Potential benefits are like shift the emphasis in medicine from reaction to prevention side. So at present, we are using the reaction side only, but in future, we are going for prevention of the disease in advance. And then predict susceptibility to disease, improve disease detection, preempt disease progression, customize disease prevention strategies, prescribe more effective drugs and avoid prescribing drugs with predictable side effects. So we can prescribe whatever effective drugs available, only those drugs, not those drugs which are some side effects. We will not choose those particular kind of drugs. And increase patient adherence. We can increase patient adherence with help of personalized medicine, because when targeting the right patient, then patient adherence will be higher, because we are providing the right drug to that particular person. With the help of this, we can improve quality of life with the help of personalized medicine. Reduce the time, cost, failure rate of the pharmaceutical clinical trials. Because initially, when we use clinical trials, pre-clinical trials, it required at least 10 to 15 years, I told you. But when we use the right responders in personalized medicine, that time period is reduced only to 3 to 4 years only. In 3 to 4 years, we can come with the new drug for particular disease, for particular patient, for particular subgroups. And revive drug that failed in clinical trials. We can also revive the drug if it is failed in clinical trials for another kind of indications, or that is already withdrawn from the market. We can revive that thing because time period is less. We can use in another indications, or you can say we can use in repurposing of that particular drug as the latest new word, and control healthcare cost by avoiding unnecessary cost when drug is proven ineffective. We can save money, or you can say that is more economic, because if drug is not proven effective, then why we use that particular drug unnecessarily? We can stop it at particular point. And social impacts of, because social impacts are very much there, because some decisions we have to take, because some potential legal and ethical questions are there. We have to provide answer to the society, because who should have access to personal genetic profile, because provide genetic sequencing to particular industry or particular body, then sometimes misuse may be possible. So who should have access to personal genetic profile? How will we protect genetic privacy or prevent genetic discrimination in the workplace and in our healthcare? So we also have provide this answer to this particular question, how our privacy and genetic discrimination will be saved, and how will we consumer use genetic information to our benefits. Means how these consumers, those who are having our information, how they will use for our benefit. Some limitations are also there. Some you can say disadvantage of limitations, because the reimbursement pathway testing is not established yet. Insurance companies or dysfunctional payment systems are there. Ethical issues are raising every day because on genetic testing and data sharing, and integration of pharmacogenomics, personalized medicines, and payer and regulatory environment is still ongoing. So you can say it is in nascent stage. In USA also, after Obama's initiative about personalized medicine in 2015, so it is now getting passed. But in India, still we are taking very behind in such cases. We're still not having a proper understanding of all these things, and we are in future we are looking for it. And clinicians are generally not educated concerning available tests, associated drugs, or outcomes, because clinicians are still having that sense of thinking about the old trial and error type. They are not going to take risk unnecessarily at present, and the response of the medication may be a result of the interaction of multiple genes. And certain pharmaceutical companies also creating some barriers, because they have mindset of blockbuster model, because they are thinking blockbuster. When drugs blockbuster means when large population of people are taking some particular drugs, then the particular drug is when grows $1 billion, then they can say this is blockbuster drug. But when we are focusing on particular subgroups or particular patient groups, then this blockbuster concept will be not there. And some regulatory environment are also very strict, and physicians habits, I already told that they are not willing at present to accept wholeheartedly. So it will take time to embrace this personalized medicines fully, when we provide information to them, when they attend some particular workshop related to this only. Continuous medical education we provide to them, so they will accept it. Next one is pharmacogenetics, biomarkers and drug labeling. So we discussed about biomarker in last slides also, that this play important role in identifying responders, non-responders about the medications. So with the help of that, we can avoid adverse events, optimizing drug therapy. And when drug labeling, we will find particular information on genomic biomarkers and that describe about the drug exposure, clinical response variability, risk for adverse events, genotype specific dosing, mechanism of drug action, polymorphic drug target and disposition, genes, So this kind of biomarker in drug labeling is very important. Obama initiative, I told you that it tracks the precision medicine initiative in US in 2015 January. That is launched by Obama, I told you, and this is providing the new research efforts to revolutionize how we improve drug health and treat diseases, empowering healthcare provider to tailor treatment, prevention strategies to individuals' unique characteristics, and certain goals of this that make health data more portable, ensure patient can easily access and share their own health information, including contributing it for research, rigorously protect patient privacy, security, choice, and support new research platform connecting research and participant as partners. So these are certain therapies like stem cell therapy that is used for personalized medicine, stem cell gene therapy, or specific cell therapy. Okay, and these are for these degenerative diseases, and many tissues and organs might be treated by transplanting either stem cells or stem cell eater material that is useful for various kind of diseases like leukemia, sickle cell anemia, Parkinson's disease, Alzheimer's disease, heart diseases, muscular dystrophy, spinal injury, diabetes, arthritis, osteoarthritis, cirrhosis of the liver, or in skin for burns. These are useful. Now, what are our role? Pharmacist role in personalized medicine. So, field of personalized medicine afford multiple opportunities to pharmacists. They have, because pharmacists, have specific knowledge, they have skills and abilities that make uniquely suited to advance the use of personalized medicine as a clinical tool. And pharmacy profession as a whole, that is being slow to embrace the concept of clinical pharmacogenetics, and now facing a clinical juncture that can potentially redefine the professional identity of the pharmacist. And there are different areas required for implementation of the clinical pharmacogenomics, that is pharmacy informatics, clinical decision support tool development, database management, development of medication use for policies and processes, logistic of genetic testing, research and clinical guideline development. Just a minute. This is some. So these are various societies and organizations which are helping to improving this personalized medicines, like Royal Pharmaceutical Society of Great Britain, that emphasized the role of pharmacists may play in performing genetic tests, and with collaboration with the local physician practitioners and counseling patient about the result of tests that either ordered their physician to purchase from their sources, and FDA's drug evaluation and research CDER that support pharmacogenomics for more than a decade for regular advice or applications, developing policies and processes for genomics and personalized medicines. And committee of the American Association of College of Pharmacy, they also recommendation for pharmacy education to include goals for knowledge, skill, and attitude of the pharmacist, and student pharmacist should aim to achieve the preparation of potential role of the pharmacist in the pharmacogenetics. So you find that in our syllabus also, we have this particular topic, because this initiatives only we should in future we should prepare for personalized medicine. Okay, and our pharmacy is constantly changing profession that require uh, to adopt these skills as scientific advance continue to transform the practice of medicine. And over the last 100 years, the profession of pharmacy evolved from dispensing model that focused on the formulation and delivery of drug to patient care model. Because now at present, we are more focusing toward the patient care model, more individualization of the patient, more individualization of the drug therapy, and delivery direct to patient care. FDA also providing some clinical pharmacogenetic implementation consortium, CPIC, that is in international working group of pharmacists, scientists, and other healthcare professionals that has developed evidence-based clinical guidelines for 35 actionable genes drug pairs. CPIC produced this 35 actionable gene drug pairs, so that is used in everywhere in future references. And over the last 2 decades, US FDA also added black box warning for several medications. Several medications like abacavir, clopidogrel, carbamazepines, they incorporated a genotype guided dosing algorithms into the product labeling and added pharmacogenetic information to the label of more than 150 medications. FDA included this genotype guided dosing algorithm into the product labeling more than 150 medications. Okay, and even for certain drugs like cineprevir for hepatitis C and mipomersen for the homozygous familial hypercholesterolemia that require pharmacogenetic testing before they can be prescribed. If we are prescribing this particular medicine, if you are going to prescribe cineprevir or mipomersen, so you required particular test if these drugs can be given to this particular patient or not. Okay, and this cost of genetic testing is continuing to decrease, and comprehensive genetic test that can be performed at less than $250 in US only. In India, that is about 1 lakh rupees or more than that. But in US, that is available in $250. And that is offered by these 7% hospitals in US. And now direct to consumer, that is known as DTC generic testing kits are available, and that readily available in community pharmacies. And the first DTC test kit that was available for breast cancer, and that is approved also, that is approved by FDA in March 2018. So pharmacists need to do certain steps they have to take for embracing this personalized medicine, adopt these medicines, like the required education and training, demonstrated competency, means they have to take time to time some refresher courses for that. Their attitude of engagement, support and guidance is required, and different kind of state-of-the-art facilities and resource and infrastructure that must be the best available, evidence of benefit, and they must do SWOT analysis of their pharmacy, if they can adopt or not, and what are different barriers they should remove them, and how they can create paths to go forward. So clinical genomics information system with help of electronic health record information, DNA test results or patient generated information, clinical trial information, all this required to create the complex genetic codes and that dictate an individual's response to certain therapies. So this kind of records are possible. So you can do some case level databases for genotypes or phenotypes, outcomes, family and medical history, environmental and exposures, and that will provide you research database, research given data, or you can say big data is available with you for research purpose and for clinical laboratory purpose and other knowledge sharing purpose. We can use this THR, data driven precision medicines, you can say. And this 23andMe is a particular company that is for the DTC genetic testing kit provider. This is 23andMe and Nivegenics, Illumina, Ancestry DNA. These are certain private companies. They provide facility to direct to customer genome sequencing to the public. So with the help of this, they can find out different kind of impact on drugs. And this is the body electronic. In this was published in Nature about body electronic, means wired for life, means certain biosensors are inserted or woven into the body that could alert people to medical problems before they become seriously ill. If there is some mutation is going to happen, means they predict and they tell you. This biosensor will predict and they tell you that this kind of possibility is there, and you can have daunting challenges. So we can overcome such kind of challenges in advance. So this kind of sensors are available in future also. So this can be in brain sensors or heart pacemaker sensors. That is possible. Or next one is lab on chip. When physician wants to initiate specific medication according to patient genotype and need only patient's one drop of blood, and that is on microchip. Then in few minutes, the SNP of the patient and suitable medication is displayed on the screen. So that's why it is known as lab on a chip. Okay. So this kind of this microfluidics or lab on chip technologies are also available nowadays. And how we can implement personalized medicine, because DNA sequencing that is improving every day and that is cheaper. In current cost is about 5 lakhs in India, and that will be expected to be around 50,000 or less in future. And every newborn can have his or her blood samples collected soon after birth, and whole genome sequencing is possible with that one blood drop, and that will then help in certain ways. What will the benefits of doing genome sequencing? That is identify diseases which is baby is going to be likely to be predisposed during his or her lifetime, and this will help in initiating strategies to prevent the development of the disease or reduce the risk substantially, and identify drugs which are likely to be toxic or cause serious side effects, so that these drugs can be flagged off from the baby's record and avoided if there be a need later. Because we have in that our list that this drug is not suitable for this particular baby, so you can flag off or you can remove from that particular list. And we can identify drugs which are more likely to be effective for diseases manifested during life and with minimum side effects, and tailor the treatment based on biological characterization of the cancer if one of the develop it later in spite of preventive strategies, and this ensure that most effective drug targeted to the specific cancer are given, increasing the cure rates and minimal side effects. So this instead of casting a traditional horoscope soon after birth. At present, what we do, we prepare some traditional horoscope, what kind of planets are there in its horoscope. But in future, what we will do? Now it is possible and prudent to get the genomic horoscope done for the baby. Now every baby will have genomic horoscope. In that genomic horoscope, what kind of disease is possible, what kind of drug he can track, everything will be available for him or her. So this personalized medicine encompasses of all sort of personalization measures. How we can personalization done with the help of certain latest techniques, latest gadgets and equipments, latest methods like use of genomics, proteomics, metabolomics. We already discussed all these things: genome, transcriptomes, protein, RNA sequencing, high throughput screening, this phenotyping screening, and imaging analysis with the help of functional MRI, computerized tomography, PET, single photon emission computed tomography, fluorescent markers, that is quantum dots. This quantum dots will be available for the future and for the imaging purpose and for detection and diagnosis of particular diseases. And radio tracers, HRLCMs, LCMS, and some theranostics. Theranostics means therapeutics and diagnostics with one particular thing. So nanoparticle based theranostics, that is deep learning algorithms, fluorescent in situ hybridization, 3D printing for drug development. This 3D printing for drug development is also very important aspect for future in drug development, for preparation of drugs as much as wanted, required for rare diseases where very low drug is required. You can prepare it at home with the help of polymers, API available with you. You can produce yourself. Similarly, as drug is available in the market, this 3D technology, printing technology, and coding holds exome sequencing, that is WES, or WGS, that is whole genome sequencing, and next genome sequencing, next gen sequencing, that is NGS, leads to genome medicines available till 2025-26. Means when we do complete next gen sequencing available with us, we can go for the genome medicines that will revolutionize completely the medicine world, that is genome medicines. Now we talk about the Indian Genome Project, that is known as the power of billion. That Genome India project aims to map the diversity of India's genetic pool, and that lay bedrock of the precision medicines to enable greater medical efficiency in terms of preventive interventions and customized treatments. And this project is funded by Department of Biotechnology, and for that 238 crore budget is sanctioned for it, and that is collaboration with 20 research institutes of very big institutes of India. And nodal center is Indian Institute of Science, and that is Indian Institute of Science Center for Brain Research, Bangalore. That will be nodal point. That will collect the all data with 20 different organizations. And this is the first step. The project will develop a reference Indian genome, that unique genetic material or sample of the 10,000 persons across India. Then during second stage, another batch of 10,000 with disease of the patient, we collect the data for that. And 3 broad categories chosen for that, that is non-communicable diseases like cardiovascular, diabetes, mental illnesses, cancer, that will be collected. And these are the the genomics industry, that is also predicted to grow at the rate of 18% in next 10 years. So you can say there is a boom in this particular industry also. And India is also expected to be a major stakeholder because of in population wise, India is the number 2 in the world. So we are having major stakeholder. And Sweden is also helping India in this particular project. They are biomolecular research and comprehensive biobanks of the quality registers are available, and very positioned as the front runner in the field of precision medicines. Sweden is very, you can say, front runner in the personalized or precision medicines, and different organizations or institutes where these genome projects are working. That is AIMS, Jodhpur, Center for Cellular and Molecular Biology, Hyderabad, Center for DNA Fingerprinting and Diagnostics, Institute of Genomics and Integrative Biology, Gujarat Biotechnology Research Centers, and IIT Allahabad, IS, Indian Institute of Science, Education and Research, Pune, that is available in your city itself, IIT Madras, IIT Delhi, IIT Jodhpur, Institute of Bioresources and Sustainable Development, Institute of Life Sciences, Mizoram University, National Center for Biological Sciences, National Institute of Biomolecular Genomics, National Institute of Mental Health and Neurosciences, NIMHANS, that is, and Rajiv Gandhi Center for Biotechnology, and Sher Kashmir Institute of Medical Sciences. These are institutes which are involved in Indian genome project. So very soon we have complete data with us. So we now coming towards the conclusion of this particular topic. So Global Genomic Center for Healthcare predicted, just a minute. This personalized medicine is a new frontier for healthcare, combining genomics with the unprecedented impact. Big data analytics, population health, and more accurate, precise, or proactive and impactful for each individual that comes under their career. So with the help of PM, we provide them the concise and proactive, accurate, impactful treatment, diagnosis, test that can provide this. And the pharmacist role is also very important. We discussed on this. Individualization therapy opens up existing new opportunities for pharmacists to expand their clinical roles and responsibilities. And personalized medicines have identified as a key and prospective approach to achieve optimal individual health decisions, and therefore overcoming the challenges of engineer better medicines. We can engineer better medicines with the help of personalized medicines. So, with that, so my sincere gratitude and acknowledgements to Professor and Dr. Ganesh Y. Dama sir. He is Honorable Principal of Sharat Chandra Pawar College of Pharmacy, Dr. Subrajit Mantri sir, and Dr. Sumit Ashok Joshi sir, all organizing team members, and all the budding pharmacists who are listening to me in Pune and all other part of our country in this national webinar. Thanks for your patient listening. I hope I provide you just iceberg deep in personalized medicine. You will more explore it. You will read more about it. And I also request faculty to provide them some projects related with personalized medicine so that they are more aware about it, and try to include in your syllabus, so they will enrich their knowledge about it. And if you have any query, I will be happy to answer about that. And I want to, because we are facing pandemic time, COVID-19, we are facing from last 1.5 years. So many people are satiated, many people affected very dangerously from it. So we should be very positive mindset at particular time. We should every day we should set some small daily goals so that we work on that and our mind is not particularly diverted. And we should carve our time in work. We should have time for relaxation. We should practice good self-care. And very important one technique we should use. As a student, we can use this Pomodoro technique. Pomodoro technique is means you should work for 25 minutes and take 5-minute break. Again, work for 25 minutes, 5-minute break. So when you apply this kind of study sessions, you will have better and better understanding, better remembering, and you will not feel tired. You will enjoy your work. And you should also schedule some get together with your friends. A lot of platforms are available. And you must prioritize your mental health. You must challenge yourself for learning. Or sometimes if we are not productive, but we should accept it. And we should avoid too much social media or news, negative talks, because this type of distraction affects us, our thinking. And we should practice healthy and mindful eating, exercise, good sleep. We should relax and we should recharge ourselves. We should recharge our battery in positive manner with a yoga or a meditation. We should practice regularity. We should regulate our emotions also. We should work for our elders' care, because at present they are very much neglecting. We should see towards them. They are the pillars of our. We should not leave them in dirt because of this pandemic. And we should develop one strategy for moving forward. And we should focus on the things we enjoy. You must participate in certain activities or online competitions, like your college, Sharat Chand Pawar College of Pharmacy, is providing a lot of opportunities to you in terms of quizzes, posters, some audio files, video producing kind of things, competitions. So you must participate in such activities at present. And you also accept what is not, cannot change. You must accept new normals. And I wish everyone stay safe and stay healthy. Thank you very much. Thank you, everyone. If you have any query, I will try to answer otherwise. Thank you.
Thank you very much, sir, for
and
sharing
you
your
can
valuable
contact me. This is my email address and my number is given. So I will happy to give you information.
Yes, sir. So thank you very much for sharing your valuable talk. So now we'll come to the questionnaire sessions. I request to all the participants kindly raise your hand so that it will be easy for me to announce your name, or directly you can unmute yourself and you can ask to our speaker, sir. So, sir, in the questionnaire session, one participant, actually he is my senior and he is from Germany. His name is Dr. Abhishek Acharya. So, sir, Abhishek, sir, you can unmute yourself and you can ask your question, sir.
Hi, Shubha. Thanks for allowing me.
Thank you.
Am I audible properly?
Yes, sir. Yes, sir.
The question is, what I presently listening is about the talk, and it's quite the area where we do research.
Abhishek sir, kindly unmute yourself. You are muted right now.
Yes, sir.
Abhishek sir, kindly unmute yourself, please.
Oh, it's not audible enough.
No, sir. Actually,
I'm
Abhishek
not
sir.
audible.
Yes, sir.
Just a minute, sir. Hello.
Sir, 1 minute, sir. Abhishek sir.
Okay. Yes, now I'm audible.
Yeah. Abhishek sir, kindly unmute yourself. You're muted right now.
Mhm.
Abhishek sir, can you hear me, sir?
Yes, I'm listening you.
No, sir. Yes, sir. Actually, I'm asking to the participant, Abhishek Acharya. Actually, he is muted right now. Abhishek sir. Okay. I think he's having some network issue, I think.
Yeah.
Yes, Abhishek sir.
Yeah, there was someone muted me, I guess.
Yeah. Okay, sir. Yes, sir.
Am I clear now?
Yes, sir. Yes, sir.
Great. The question is what we see in personalized medicine is the incorporation of stem cells, because most of the time I didn't find it in the presentation. However, this falls in two categories. One is gene therapy and another is cell therapy. Most of the topic which today covered, I can see it's a one part of personalized medicine. Why I will also take a moment to explain, because if we see 5 people, probably 5 people's choice of listening music are different. It's because every individual's choice, and they have different response to different music. So was our body system. What happens is, when we look at only gene expressions or metabolomics, we may miss tissue specific physiology. Whereas stem cell actually helps us to create those specific cells. For example, if a person's getting a heart attack, and we are trying to give gene therapy and trying to find out some mutations if they're at all, and then we can correct it, probably we have to first study what's the response to the cardiomyocyte of the patient. That's why this personalized medicine will be incomplete if we only consider the gene therapy, not the stem cell incorporation, because often we see the challenge by just considering the global transcriptomics and proteomics, which do not give us tissue specific changes. There are many examples. We see the spinal muscle atrophy, which is very common. This is very young infants they suffer with, and there is one therapy you can study their SMA gene and you can treat from the mark, and the drug is Jolisma, and that's how the gene therapy works. But when I go to real personalized medicine, when I have to understand the individual, then I must look into its tissue homeostasis or the cell homeostasis, and then only I have to customize a drug or probably a gene therapy. I would like to know what's the honorable speaker thinks about it, and maybe he can throw some light what we see, how we can incorporate stem cells, the individual stem cells, and then gene therapy, so that combined they will be called as personal medicine. Thanks, Manu, and Manu is Subrajit. He's my junior, and thanks for organizing this amazing talk. Thanks.
Thank you so much. You are also elaborated in wonderful manner, because stem cell, I just only give one brief introduction about that, because that is useful in various diseases. When stem cell, because this is used in stem cell banks, And whenever somebody is suffering from any particular kind of this diseases, that time this regenerates cells and useful in particular diseases, cure from that. I think I'm aware about this much because it is a very vast field.
Yeah.
Thanks for your.
I think, sir, Abhishek sir also working under this personalized medicine. Actually, I talked with him also once, and he said that he's also working under this personalized medicine. Thank you, Abhishek
Because
sir,
there are
for
so
asking
many,
a valuable question.
there are so many aspects. That is one
Yes,
aspect
sir.
of
Yes,
that.
sir. Yes, sir. Yes. Thank you, Abhishek sir, for your asking a very nice question, and also you're interacting very nicely with us. Thank you very much, Abhishek sir. So now I will come to the next participant, Upasana. Kindly unmute yourself and you can ask your question, please. Upasana, madam.
Good afternoon, everyone. So my question is this personalized medicine is only the study, or it's also a research part that we study about the genes? Like you mentioned that 10,000 diseasable persons and 10,000 communicable diseases person are tested, and the difference of gene is studied, and that's why the personalized medicines are given. But is it possible to check or modify the genes and that and collect the research that yes, this gene is really happen for that particular disease?
Yes, Upasana, very nice question. As you know that we can change mutations, and because if we change mutations or SNPs, then we can we can replace with the healthy gene. That is, and this is not only data, this is implementing everywhere in US and in England, Sweden, India. We are using this genetic information, genetic sequencing every day. Now we are using it for benefits. So it is just not only concept, but it is a reality, you can say. In future, genomic medicines that will become in 2025, about this will be the case. You will see that this medicine will be completely replaced in future. There will be a complete revolutionize, you can say. These quantum dots are coming, or different newer drug delivery systems that is only target specific, very low dose with very less side effects. Complete individualization will be there, you can say. So this is possible, not only just research or something. Are you okay? Is it okay?
Yeah. Yeah. I think Upasana madam muted.
Yes, this is totally okay. And very nice interactive session, sir. Thank you for all that knowledgeable things you give us to us.
Thank you so much.
Yeah. Thank you, Upasana. I think there is no question from the participant. So once again, I'm requesting to all the participants, if you have any question, kindly type in the question in the chat box. Or yes, sir, I got one question from one of the participant, Dr. Ganasyam. Actually, he is asking that what is the role of personalized medicine on autoimmune disease?
Yeah, personalized medicine role in every kind of disease, but we have to know that that faulty gene. If we come to know and we detect it, we test it, and after that we can use that personalized medicines in every disease, not only in autoimmune, but in all kind of diseases we can use.
I think Dr. Ganasyam sir, you got your answer. So, sir, we have only this participant question. So now I'll move towards Ms. Vaibhavi Kapatkar, madam, to give the summary of this session. Kapatkar, madam, it is over to you.
Okay. Thank you, Dr. Mantri. And thank you, Dr. Kamal Rathore.
Thank you.
I and everyone here will believe that it's a vast and exciting topic on the scope of personalized medicine. To be very frank, when we decide to take up the topic, what to take up for webinar, and when this topic comes up, it's an overwhelming topic. Yes, if you see the personalized medicine, more business in today's and in the future world, wherein, yes, developing new therapies and commendable milestones we can achieve with this personalized medicine. You have rightly said, sir, right drug at the right dose and in the right patient is very much required. And yes, one dose doesn't fit into all also. Everybody's therapy is nobody's therapy. That also
Yes.
comes very much true through your presentation, sir. Personalized medicine, yes, it has the ability to offer the right drug to the right patient for the right disease at the right time with the right dosage. Very well presented, sir. And you have thrown light on many aspects today. Many strategies to keep ourselves motivated also during this pandemic. How this changing of mutations and SNPs are going to help us with the scope of personalized medicine has been very well discussed, sir. I would also say, sir, when you discussed about pre-screening, wherein you said that we should allow the clinical trials to be smaller, faster, and less expensive. And when we compare with the today's scenario, during this pandemic, this has actually been taking place, and that was the reason why these vaccines came into the market so fast. That was all the reason of the clinical trials being small, faster, and less expensive also. Yes, in the aims and scopes, you have covered up the topics on implementation of genetics and then advances in molecular diagnostics, national sequencing programs, impact of precision medicine strategies on drug development as well as in healthcare delivery, sir. What sounded me interesting was the pharmacist's role in this personalized medicine, sir. So I was very much keen to understand the pharmacist role, which also you have specified that through his knowledge, skills, and abilities will help in the advances of this personalized medicine. His engagement attitude, of course, will count a lot. Evidence of the benefit, support and guidance, resources, and infrastructure. I agree to Mr. Acharya also what he said on elaborating the stem cells use for the wide scope of personalized medicine. Sir, it was a great session, very informative session. I am expressing my vote of thanks to you, sir.
Thank you.
Expressing my gratitude to your sir for your presence and contribution, and also taking out time from your busy schedule to grace this event, and yes, making this webinar a very great success. Thank you, sir, for inspiring and encouraging us with your words on this special day. I also must thank to the organizing team and to the participants for making this webinar a real great success. Thanks a lot, sir. Thank you.
Thank you, madam. So
Thank you.
I would like to request our principal, sir, Dr. Ganesh Dama, sir, to conclude this session. Sir, it's over to you, sir.
Yes, thank you, Dr. Kamal Singh Rathore, sir.
Thank you, sir.
Very much thankful on behalf of our webinar team and all the participants, those who are being present with us for this today's session. And really, you have touched the each and every part. What from your side to cover this topic, and I think that whatever the participants, they have raised their questions and all, you have clarified their doubts also. And hope if they if again they are having the doubt, you have shared your cell phone number also. They will contact with you also, so that you will make yourself available for them. Those
Yes, sir.
participants. Actually.
Always, I'm always available for.
Yeah.
Yes,
yeah.
sir.
And you have touched everything part from your side, because personalized medicine actually number of things that you have been covered already. Madam has been summarized each and everything. I will not repeat all these things again because each and every I want to get it trained my faculty also, so that they can come forward and to, because every time I am there, but they should also get it trained from for each and every part, because Mantri sir is expert in another thing. Faculties
Yes.
are expert from the other side. So they have been summarized here. So on behalf of our webinar team and on behalf of my institute, Sharachandra Kaur College of Pharmacy, which is affiliated to Pune University, I'm thankful to all the participants, and particularly very much thankful for your talk or for your webinar from Rathod sir. Very much thankful for
Thank
your
you, sir.
university, for your colleague also. Thank you, sir. Thank you very much, sir.
Thank you,
And
sir.
this is your
Thank you so much.
certificate of recognition. I will post it on your
Sure.
mail so that you can
Okay, sir.
receive that certificate. This is an honor to us, actually. It's not
Okay.
honor to you, but it's an honor to us
This
to
is very,
issue this certificate.
this
Yes,
is
sir.
very
Thank
beautiful,
you.
very nice.
Yeah. Yeah. Thank you. Thank you very much, and thank you. Thank you all
Okay,
the
sir.
participants for this for your contribution or for participating for this webinar also. Thank you. Thank you for everyone.
Okay, sir. Thank you so much.
Okay, sir. Okay, thank you. Okay, sir. We'll do the meeting. No problem.
Thank you, sir.
Okay.
Okay, sir.
Thank you. Thank you, participants.